So a little bit about me. I'm a pharmacist by profession. I went to University of Auckland, and then I worked as a community pharmacist and got my registration in New Zealand. And then I moved to Australia, and I continued working as a community pharmacist throughout my career. My thesis was on nanomedicine for brain cancer, and I graduated last year. And then I continued working as a postdoctoral research fellow at the University of Queensland at School of Pharmacy. And my work is funded by Cure Cancer and also by Brain Foundation grant. And previously my PhD was funded by Tour de Cure. Yeah. So glioblastoma, we've heard a lot about AML, so it'll changing of tracks a bit. I'll talk about brain cancers and specifically glioblastoma, which is quite a malignant brain tumor. Patient survival is less than 5% for 5 years, and average survival rate is about 14 months, which is quite abysmal. And there's three kind of reasons why glioblastoma is so tricky. It's really deadly. Often it's diagnosed quite late, so patients are quite, it's quite advanced by the time it's diagnosed. Well, we compare it to something like leukemia or breast cancer. We have now really good therapies available, but brain cancer and glioblastoma, 1 out of 10 patients are likely to survive for 5 years. And the treatment is quite expensive as well. It costs more than any other cancer treatments. And then, this is, 2014, a bit old, but, less than 3% of funding goes towards brain cancer. That might also be because less patients are diagnosed with brain cancer. But so since early 2005, the mainstay of the therapy was surgery, followed by radiotherapy, and then often, surgeons will plant a carbamazepine wafer implant. It sort of slowly releases drug over a few days, and that is dependent on how the tumor was debulked from the brain. Then in 2004, bevacizumab came. It stops blood. It's an anti-angiogenic drug, but it hasn't really improved survival. It does improve quality of life, though. And then a game changer drug came, which is called temozolo-mide. It's an oral drug, and it improved survival. So patients' survival went from 7 months to 9 months to about 14 months. But it has issues as well. It has a very limited brain penetration. So from blood to brain, 20% of the drug goes into the brain. And then we have a lot of dose-limiting hematological toxicity as well. It's got a short half-life. It's quite unstable in plasma, so you need to do multiple doses. And then even with that, about 50% of the patients will start ending up, not responding favorably to it. And obviously, that is mainly due to blood-brain barrier. A lot of chemotherapy drugs are excellent, and we know that they will work really well against brain cancer, but they're not able to cross the blood-brain barrier. And there's a lot of exciting work that's happening in this space, from immunotherapy to improving the blood-brain permeability. My work has been focused on nanomedicine and specifically silica nanoparticles. So silica is ubiquitous. You have it in so many things, like from the toothpaste that you eat to the water to potatoes, carrots. But interesting thing about silica nanoparticles is it can be modified to different shapes and sizes. And in fact, it has been used in humans as well. It's very limited. It's still an emerging material, but there have been starting to be some phase 2 trials as well. In fact, for brain cancer, there was a clinical trial which showed that it could be used for diagnosing brain cancers. So this was a Cornell dots. They're like 10 nanometer silica nanoparticles, which had a sci-fi dye loaded into them for diagnosing brain tumors. But primarily, they've been used for 3 sort of things: drug delivery, photothermal or plasmonic therapy, or diagnostics. So for glioblastoma, though, we'll need sort of, for any drug delivery agent, we'll need sort of 3 main criteria. It needs to be stable. It needs to deliver the cargo before degrading and also have an optimum size. So small enough that it can penetrate the blood-brain barrier, but not so large that it's not so small that it will just get renally cleared. And then the shape and functionality should have some ability to modulate it, to target it. Unfortunately, current nanomedicine is dominated by liposomal formulations and other organic materials, which are very difficult to synthesize in small size, or if we do synthesize them in that small size range, they suffer from stability issues. And that's where my project comes in. During my candidature, I developed these ultra-small silicon nanoparticles. The synthesis is quite simple, actually. It's made in room temperature. We start with like water, a catalyst, and a surfactant, and that leads to micelles self-assembling. And then we add a silica precursor to it, which decorates around these micelles. And I also use a pore expander in the oil phase to make the pores quite large. And then we calcine it, we just burn it. So there's no, wash it and burn it. So there's no other reagents left except for the plain silicon nanoparticles. And they've got large pores, so we can carry a lot of cargo into them. And they're quite small, so 30 nanometer size. This is the electron microscopic image of these nanoparticles. So after developing these nanoparticles, I tested them and to see if we could deliver temozolo-mide. In my previous project, I've also worked on delivering doxorubicin. So yeah, as I mentioned, temozolo-mide has these formulation issues, and we looked at whether we can deliver it better. So the hypothesis was whether we can enhance its delivery by having some targeting moiety attached to it. So the targeting molecule that I selected was lactoferrin, the receptors of which are found really highly in the blood-brain barrier and as well as the glioblastoma tumor. And then we pegylated it as well to reduce the efflux issue because it was more likely to stay inside the brain. So using a really interesting chemistry, we attached the PEG onto the silicon nanoparticles and then pegylated them as well and loaded the drug into the large pores, or alternatively, attach sci-fi dyes and stuff, and then attach the lactoferrin nanoparticles on lactoferrin compound onto these nanoparticles. So the lactoferrin will allow it to target the brain as well as the glioblastoma tumor. So we started with some in vitro work, looked at some blood-brain barrier, in vitro blood-brain barrier models, and we found something really interesting. We found that the silicon nanoparticles on their own can actually modulate claudin and zona occludin, which are tight junctions found on the blood-brain barrier. But when we add the lactoferrin on the nanoparticles, it stops doing it. It enters through a different route. It does not modulate the tight junctions, but it is receptor taken up by the receptors of the lactoferrin. This work, Cipherid modeling was done in UNSW in Professor Maria Calvalar's lab, who's my collaborator. And then we looked at whether it would enter the 3D spheroid of glioblastoma, and it was really cool. We found that lactoferrin coated and pegylated nanoparticles had really good penetration into the tumors. And then we looked at seeing if we could reduce efflux. So using this similar in vitro blood-brain barrier model, this is a HCMD3 cell line, which is a human cell line of the blood-brain barrier. And we found that if we put a nanoparticle at the top with the drug, and we calculated how much of it goes from blood theoretically to brain, or the efflux, which is from basolateral to apical chamber. And we found that the efflux ratio of the tumours, it's not labeled here. So this is the control temozolo-mide, the black, and then you have the nanoparticles, which is with just PEG and then lactoferrin. So it showed that the efflux also decreased, the efflux ratio. Compared to pure temozolo-mide, which is good because we want the drug to stay in the blood, in the brain, not come out. And then also the cytotoxicity was quite enhanced as well. This was done using flow cytometry. And so, we had really good early apoptosis and late apoptosis in both GL261 and U87 cell lines of glioblastoma. And then lastly, we did a sort of proof of concept study to see if these nanoparticles would enter into the brain of the mice. So these were healthy mice, and we looked at giving them these formulations, and we found really straight up that in the brains of the mice, after 1 hour, you could start seeing the nanoparticles accumulating in the brain. But when it was had lactoferrin attached to it, which is a targeting moiety, we started seeing a really high signal in the brain, and within even 1 hour, and then it slowly started decreasing. So with adding the lactoferrin, we found that we could accumulate faster into the brain the nanoparticles. Obviously, this was without the drug, so we need to do more studies to see whether that equates to efficacy as well. So yeah, we know what works. We know that this sort of formulation, the ultra-small silicon nanoparticles, can be used as a delivery system for temozolo-mide and doxorubicin, the data of which I haven't shown yet. Doxorubicin is an interesting drug because it cannot go into the brain at all. It's a very hydrophilic drug, whereas temozolo-mide, some of it does go into the brain. So this formulation can be used for both hydrophilic and hydrophobic small molecules, but also large molecules, which we haven't tested. But I hypothesized that it would be able to be used for large molecules as well because of its large pore. You can see the large sort of porous structure of it. This is about 7 nanometers in size. So it's quite a porous structure. And then we can coat it with different things on it, depending on what we need. So in my case, we have done lactoferrin and pegylation, but we could attach as easily any other targeting moiety on it as well. Yeah, and we looked at 2D and 3D cell cultures of both blood-brain barrier and to show that it does have enhanced permeation into the brain and possibly better therapeutic response as well. We have also some data, which I haven't shown yet, that in 3D glioblastoma models and blood-brain barrier models, the nanoparticles actually really penetrate really fast into the brain. And some preliminary in vivo data. So next, we're looking at seeing if we can have better response in 3D tumor models, and especially in vitro, and using a technique called donuts that was developed by Professor Maria Clavellar's lab to see if because when we do it in 2D models, we lose a lot of the spatial and temporal information for nanoparticles. So that is the next step. And then also looking at in vivo spontaneous glioblastoma models. So often when we insert a tumor into the mice brain, what ends up happening is you can affect the integrity of the blood-brain barrier. So with the spontaneous model, the fidelity of the blood-brain barrier remains intact. There's no artificial disruption to the blood-brain barrier. And so that's really and it's honestly a better model for looking at biodistribution. And then next we'll be testing efficacy in patient-derived xenografts. Obviously, glioblastoma, just similar to other cancers, is quite heterogeneous. So patient-derived xenografts will be a very good approach to testing this. So the work of this is going on at the moment. So hopefully we see something interesting. Yeah, I'd like to thank my own group as well. I work with Professor Puppet in University of Queensland, and then also Professor Cavallaris and Professor Chris Siriski from AIB, and we've been really helpful in doing a lot of these studies, especially like animal and 3D syphilis models. Thank you.